In addition to regulation of gene expression at a chromatin level, and then at transcription level, post-transcriptional level, there is an additional level which is found after the proteins are synthesized. That is called post-translational modification of the protein. So very simple example of post-translational modification is after the synthesis of hemoglobin protein, the heme group is added to it. Heme is an inorganic part of the protein. So it is added, it's a modification that is done to the protein to make it functional. Just imagine if the protein does not have heme group, what type of activity of hemoglobin would be. So basically none. So because that's the whole purpose of hemoglobin, to carry oxygen. So if there is no heme group, there is no place where the oxygen will be carried. So lots of other modifications are done to the proteins. Another example is in antibodies. Antibodies are glycosylated. The addition of carbohydrate groups are done to antibodies, so that makes them soluble. So proteins are able to be transported in the blood. So these are the two kind of simple examples. What these modifications do, they make those proteins functional. Proteins are able to move. They are able to be they are able to be localized, or they have some more affinity for some molecules or the others. So there are several different reasons for these type of modifications. The most modifications that are done to the proteins are the addition of phosphate groups, which is called phosphorylation. So the addition of phosphate groups to the proteins phosphorylates them. And what phosphorylation does to the proteins is either proteins are activated or proteins are inactivated. So some proteins get activated, other proteins get inactivated. It is done with the help of these enzymes called kinases. Kinases are those enzymes that phosphorylate proteins, and phosphatases are to remove those, to reverse the addition of phosphate groups to the proteins. So and there are so many genes in case of humans that are responsible for that. In case of kinases, there are about more than 500 genes in case of humans. More than 500 genes are related to producing kinases. And in phosphatases, this is about 150. Around 150 genes are related to phosphatases. So that means they are fairly significant part of the genome. They are significant part of how things function. Example of these type of phosphorylations is in the form of transcription factors. Transcription factors that initiate the process of transcription can be modified with phosphorylation that will make them either turned on, they will be activated, or they could be turned off by phosphorylation as well. Other examples are in cell cycle proteins, cyclin proteins or CDK proteins, cyclin-dependent kinases. So these are all examples that indicate how important these type of modifications are for proteins to make them functional. Protein degradation, after they are utilized, after they are synthesized and used inside the cell, also follows a very similar pattern of degradation, like in case of messenger RNAs. Enzymes are involved in the degradation of proteins or nucleic acids. In case of in those proteins that have gone beyond their utility, or have changed, or have been broken down, or have been synthesized with the wrong sequence of amino acids, those are all identified, and they are tagged with some small proteins called ubiquitin proteins. Ubiquitin proteins are small proteins. They are less than about 70 They are less than about 76 amino acids long, and they're small proteins. They are called ubiquitin proteins because ubiquitous. They are, very abundantly found in the genomes. So proteins that are meant to be degraded, they are targeted by these proteins, which are very small proteins. The process is called ubiquitination, and that process of ubiquitination tags those proteins, and it is taken to another shredder-like complex of protein called proteosomes. Proteosomes are the ones that identify those proteins which are tagged with ubiquitin, and then they are degraded. How do ubiquitin proteins know which protein to target and which protein are meant to be destroyed? It is actually a pretty significant regulatory process that is followed inside the cell. So this is not the place to really discuss it. But the enzyme that does this job of adding those small proteins to the protein that need to be degraded are called ligases. So ubiquitin ligase is that protein that identifies those and tags them to tag those proteins that are meant to be degraded. Ubiquitin proteins so there are about 600 uh, ubiquitin ligases, that are meaning that there are that many genes that are found in the human genome, and there are about 9,000. So the number of genes for this ligase, ubiquitin ligase, is about 600, and these ubiquitin proteins are tagged to about 9,000 different proteins. So big part, which is about 40% of the human genome. So about 9,000 of those proteins are degraded by using this process. So it is a very significant process followed inside the cells. Um, Protein that needs to be degraded. So this is that substrate, that protein, and ligase adds one ubiquitin protein at a time, and it makes a long chain. So it is kind of a polyubiquitinated protein. That polyubiquitin protein tail or chain is identified by proteosome. Proteosome is it's a large complex protein. After it attaches the substrate, which carries those tags of ubiquitin protein with it. So the first thing that happens to the proteins is those ubiquitin proteins are removed from it. The protein goes inside the proteasome, and proteasome chops them up into smaller fragments of amino acids. Those small fragments of amino acids are further degraded, and they could be recycled with the release of amino acids, and can be reutilized inside the cell. So this is another level of regulation. So when the proteins are not needed, or if something happens to the protein at the time of their synthesis, so they can be removed, instead of they becoming part of the cell and perform function which may not be the normal function. There may be faulty proteins, so those have to be eliminated. So that takes care of those proteins.